And so now it's my great pleasure to introduce our keynote speaker, Deanna Bianchi. She's the director of the National Institute of Child Health and Human Development since 2016, 2017. She's probably the person that epitomizes what we're trying to do here at Stanford the most. She was undergrad at Penn and then became a medical student here at Stanford. In fact, I think she worked with some of the faculty in the room when she was here. She was noting actually that the medical center has changed just a little bit since she was here. Understatement. Then went on to a pediatrics fellowship at Boston Children's. Then had an illustrious career at Tufts, where she really started what was now the MCHRI Institute at Tufts, and then went to the NIH, to Boston and NIH after that. She's gotten so many awards, including the National Academy of Medicine in 2013, and notable of all the awards she's gotten, the Sterling Lifetime Achievement Award for alumni at Stanford was given to her 2 years ago. I think that really epitomizes her dedication to the field and also to Stanford. And so we really, I think for me, some of the most profound publications that have come out of her lab include the identification of cell-free DNA as better than using traditional therapies for detecting fetal anomalies. And so she still runs a lab at NIH, as well as directs and has transformed the Institute. And so I just want you to join me to welcome Deanna Bianchi today.
Thank you so much, Tony, for the kind introduction. Can everybody hear me in the back? Like Cynthia, I wake up in the morning thinking about science and I think about money, but I think about it from a different perspective. And it's really an honor and privilege to be thinking about how to most effectively spend your tax dollars. So I used to think, even before I went to the NIH, when I was paying my taxes, I was paying my taxes to the NIH and to the National Park Service. But seriously, we at NICHD have a budget of $1.5 billion. That's a lot of money. I had to get used to saying billion instead of million. And so, when I think about money, I'm thinking about how can I plant the seeds for the young investigators? How can we support them, keep them in the pipeline so that they will go on to have impact and achieve successful careers? How can we fund the most impactful science? How can we bridge people who are temporarily between grants? So all these kinds of things, it's truly an honor and a privilege to be in that position. It all began, the research I'm going to tell you about all began here at Stanford. And I did want to point out that, yes, I am on Twitter. So feel free to tweet, and NICHD will then retweet the kind comments. If you have something that you're not happy about, come to the question and answer session when I'm happy to discuss that. But I will be talking about some of the work that started here and has continued in my laboratory in the Genome Institute, because as a director, there's a conflict of interest. You can't be essentially giving resources to a laboratory in your own institute. So it was a very easy decision for me to have a laboratory in the Human Genome Institute, which is kind of like, for me, it's the icing on the cake. So we'll get started. How many people are familiar with the All of Us program? So it's a huge signature program at NIH, really Dr. Collins' vision. We're enrolling 1 million people of very diverse backgrounds for a variety of studies, and hopefully will impact upon their health. It's one of the most ambitious efforts undertaken at the NIH. Anybody know how many children have been enrolled in All of Us? 0. Yeah. So I've been at NIH for 3 years, and we've been trying to get children enrolled. So far, we have not been successful, but we did find out that over 3,800 pregnant women have been enrolled into All of Us. So we are trying to leverage that population. Unfortunately, we still can't link the babies to those mothers, but we are linking those mothers to something called PregSource that is funded by NICHD to try to get more information on the experiences of these women, and in particular the medications that they're taking. So when we talk about personalized medicine or precision medicine, at least at the NIH, we're not yet talking about children. But my interest in fetal and pediatric personalized medicine goes way back. And I will be talking, this talk will be mainly about the science. At noon, you can ask me anything about NICHD. We're very happy that we've published our first strategic plan in 20 years. So the last strategic plan was in 2000. There are a number of research themes that very significantly align with the work that you're doing here. But if you go to the back to the 10 aspirational goals, one of them is personalized medicine for children. So I have a personal commitment to research in Down syndrome that actually preceded time at Stanford. The letter on the left is in French. Those of you who can see the Mademoiselle can see that part. When I was in college, I wrote to Jérôme Lejeune, who was the professor in France who discovered that Down syndrome, the basis of Down syndrome was 3 copies of chromosome 21. And apparently back in 1973, I wrote to him and asked to work in his lab, and he very kindly replied in French that, your French is great, but unfortunately I don't have any space in my lab until the next year. And I couldn't take a semester off to do that. I have that letter framed in my office at NIH. After doing cytogenetics work as an undergraduate, I came to Stanford, where I met the late Howard Kahn and the late Len Herzenberg, who encouraged me to come work in the Herzenberg lab. And I don't know if Lee is here, but certainly Lee is still here. And they told me about their son, who is in the picture on the right, Michael Herzenberg, whom Lee gave birth to. It all originates from a story, a real story and a real person. Lee gave birth to their son Michael when she was only 29 years old. And so Lee and Len were very interested in developing a non-invasive prenatal test to detect Down syndrome because they were completely unaware until Michael was born that he would have Down syndrome. In fact, even when he was born, there were no chromosome tests at that time. That was in 1960. And so they made the diagnosis from a clinical exam. Later on, they confirmed that he had Down syndrome, and he's still alive today and occasionally has worked in the laboratory here at Stanford. Well, their charge to me was to use the fluorescence-activated cell sorter that had been developed by the Herzenberg lab to isolate intact fetal cells from the blood of pregnant women. And that work on the left led to the realization that there were fetal nucleated erythrocytes circulating in the blood of pregnant women, and that you could, at least with the technology at the time, use fluorescence in situ hybridization for the Y chromosome to show that there were male nucleated erythrocytes circulating in the blood of pregnant women. There weren't enough of them at the time to use that for a prenatal screening test. And subsequently, we learned with animal models that those cells were not in the peripheral circulation for very long. They actually migrated to various maternal organs where they participated, not necessarily the nucleated erythrocytes, but other pregnancy-associated progenitor cells would migrate into the maternal organs and played a role in repairing those organs. And that's a whole other story called the microchimerism story. But moving from intact cells to cell-free DNA over time and with the development of technology, it turned out that it was recognized that there were large amounts of cell-free, quote, fetal DNA, which is actually placental DNA, in the blood of pregnant women. And you could actually sequence those DNA fragments that are in the blood of pregnant women and map those fragments to various chromosomes in the human genome and develop mathematical ratios. And that this screening was so sensitive that you could use that to actually detect various human chromosome aneuploidies. The graph on the right shows you over time this testing first became available clinically and commercially in 2011, and I do want to acknowledge the fact that Steve Quake from Stanford played a major role in this, as did Dennis Lo of the Chinese University of Hong Kong. the testing increased astronomically and subsequently led to a significant decrease in the number of amniocenteses that are being performed. So I've been very fortunate to see from my time as a Stanford medical student to have all of the issues associated with trying to extract these rare fetal cells, to see it come full circle, where it has changed prenatal screening, the positive predictive value of a test that suggests that the fetus has Down syndrome is on the order of 90% for a general population of pregnant women, compared with the standard of care where the positive predictive value is 5%. So it's really been an evolution, and as the cost continues to come down, it will become first tier for genetic screening in pregnant women. It's had some consequences, including with regard to training. It's very hard to learn how to do an amniocentesis these days because so few of them are being performed, but that's a story for another day. I'm not going to go into cell-free DNA screening, but I did want to point out if you want sort of a one-stop review of what has happened and what is being tested for, this review that I wrote with Rosa Chu in the New England Journal of Medicine published last year, it is still very much up to date, so let me send you there. Well, for us, it was never only about screening or diagnosis. As a pediatrician, it was also very much about treatment. I've been very interested in fetal treatment from a surgical perspective, as well as a medical and a gene therapy perspective. And even back as far as 2012, I was talking about the concept of fetal personalized medicine. And that's where we have to go. That's why an institute such as this one and others is going to play a major role. The earlier, the better. That is really a very important theme. As I said, the focus so far of precision or personalized medicine has been largely on adults, less so on children, and certainly not on fetuses. So what if we're going to try to improve the lives of people who have Down syndrome? What are some of the challenges? And we recently reviewed all of the clinical studies that have been done. This paper came out last week in Trends in Molecular Medicine. There have been over 30 promising preclinical studies in mouse models of Down syndrome, and there have also been 26 completed human trials. Although they work great in mice, they have largely not shown any improvement in humans, so very frustrating. And some of the challenges include the fact that the trials are small. So the largest clinical trial has had 350 people in it. There's potential inter-individual variability. We know that exists at the cellular level, and I'll show you that at the end of the talk. There's certainly a lack of standardized outcome measures, both in the mouse models and in the humans. And it's well known when you're dealing with people who have intellectual disabilities that there's a placebo effect. Just bringing these kids in, paying a lot of attention to them, testing them, giving them intellectual stimulation improves them. So there is definitely a placebo effect. And for safety reasons, a lot of the interventions have been at the adult stage or at the adolescent stage. And we would argue that is too late. So our overall hypothesis, as I said, is the earlier the better. And in our laboratory, we are testing the hypothesis that prenatal treatment given to the pregnant woman will result in improved fetal brain growth, connectivity, and ultimately improved neurocognition. The goal is really to increase IQ by 10 to 20 points, which will significantly improve independent life skills. And we know that if you look at this timeline between conception and birth, there's a tremendous amount of development that is going on in the euploid fetus. And actually things, you don't really notice much of a difference early in gestation, but by the third trimester, you begin to see the drop-off in terms of growth, particularly in the cerebellum. So the hypothesis is that if we were able to give the mother some sort of treatment, that we would bring that particular fetus's brain growth up to typical development. So we started with using transcriptomics to develop novel therapies. We had a lot of experience looking at cell-free RNA in amniotic fluid. And unlike cell-free RNA in the maternal plasma, the cell-free RNA in the amniotic fluid is nearly 100% fetal. And as we began to analyze this material, we realized that really the fetus is changing almost on a daily basis. So first of all, you need to specifically control as far as gestational age goes, but you could study, if you had access to the amniotic fluid, you could study women at serial points in time. And so particularly women who had polyhydramnios for some reason or not, who are getting therapeutic amniocenteses, were very helpful for this research because we could study them. It was discarded material anyway. And if anybody's interested in the gene sets that we have publicly available on GEO, we have matched case and control gene sets, analyses of our transcriptomes from a number of fetuses with different both chromosome abnormalities such as trisomy 18, 21, Turner syndrome, as well as certain exposures, so obese mothers versus lean mothers, and fetuses with twin-twin transfusion. So if you just go to GEO and type my name in, you'll be able to get access to those sets. Fairly early on, we realized, however, that there were a number of transcripts in the amniotic fluid, so cell-free RNA transcripts that mapped to the developing fetal brain. So an amniocentesis, while giving you information about the fetus's chromosome status, also gave you information about the developing brain. And we thought that was very, very intriguing. And we began to notice differences between, say, fetuses that had trisomy 21 or trisomy 18 and pathways and how the genes were influencing development versus via these pathways. And as we began to analyze things further, we realized that each fetal chromosome abnormality actually had a unique constellation of pathway abnormalities. And there was very little duplication, actually, between, say, a fetus with Turner syndrome and a fetus with trisomy 18. And some of the highlights included the fact that, for example, in Down syndrome, those fetuses had pathway abnormalities suggesting oxidative stress, abnormalities, decreased cell proliferation, increased neuroinflammation, and that was very different, say, from a fetus with Turner syndrome that has abnormalities with cholesterol, signs of autoimmune disease, even in the second trimester, and fascinating because people with Turner syndrome have coarctation of the aorta. You even see signs of perivascular tissue remodeling at 15 weeks of gestation. So we knew we were onto something here and thought that, well, maybe we could put these dysregulated pathways to use in identifying novel therapies. But it's not easy because you have a number of concerns dealing with pregnant women. And wearing my other hat now at NICHD, we're very aware of the reluctance to study drugs in pregnant women. And that is why one of our themes in our strategic planning process is to do more research into the safety and efficacy of drugs and devices in pregnant women as well as young children. So obviously we're all concerned about safety and the fact that we have two individuals that we are affecting with the medication. We've got the mother and we can't harm her, and we have the fetus. We don't want to cause additional problems for the fetus. And then we have two barriers to cross in terms of trying to improve neurocognition. So we have to get across the placenta, and then we have to get across the blood-brain barrier. So it's not easy. So we have a very ambitious set of experiments that really involve the human clinical experiments that are shown here, trying to figure out what are the best ways to evaluate the effects of therapy on actual children. In vitro studies that are identifying candidate therapies and then testing them on living cells and validating both the safety and the appropriateness of using those drugs in pregnant women, and then running clinical trials on mice, though preclinical trials, to see how does the proposed therapy affect the mouse model, and then going back to humans to eventually get to a human clinical trial. So when we started this, nobody was really working in this area. Most of the research in Down syndrome is at the later end of the spectrum because people who have Down syndrome invariably get dementia and a type of Alzheimer's disease. So most of the efforts are there. Nobody was really working in the prenatal part. So we initially were trying to figure out what's the best mouse model, and we compared the 3 most commonly used mouse models. Then decided what are the best endpoints where there are significant differences between typical and atypical mice. Then we analyzed the integrated transcriptomes, looking for common pathways that were dysregulated in both mice and humans. Then we screened the candidates in human cells and performed, so far we've done 1 round of experiments with 1 candidate molecule. So for those of you who are not familiar with the mouse models of Down syndrome, the 3 most common ones, first of all, there's the workhorse TS65DN, which has a free trisomy, but it also has, so first I should say that human chromosome 21, it does not have an exact equivalent in the mouse. So if you're going to map it to mouse genes, it's got primarily mouse chromosome 16, a little bit of 17, a little bit of 10. With TS65DN, it has quite a bit of 16, quite a bit of 17, but in fact it has too much of 17. It's got a number of genes that are triplicated that have nothing to do with human chromosome 21. And most significantly, there's triplication of ARID1B, which is a known gene that is involved in autism. So TS65DN is the most severely affected mouse model and has a lot of similarities with Down syndrome, but it also has a lot of genes that are not relevant to the human situation. Also, in TS65DN, the males are infertile, which is similar to the human situation. But if you're evaluating prenatal therapy, you want to have a male that's affected. You want to be able to breed the mice with a wild-type female because you want the mother to have a typical pregnancy experience and to be able to foster the mice in the way that a euploid mouse would do. With TS65DN, the only way you can propagate the extra chromosome is through the mother, and those mothers have very poor reproductive performance, and they neglect their pups. We have some really interesting videos where it's the father who's moving the pups around in the cage. The mother's really not that interested. So it's not a great model for us, but it is the workhorse, and it's sort of the gold standard in the literature. TS1CJE is a milder mouse model. It doesn't have all of these extra genes. It does have monosomy of the area where a translocation has occurred. So that's its limitation. However, the males are fertile. So we can pass the phenotype using the father. And then the one that everybody was like crazy about was duplicated 16, where there's actually duplication of all of the relevant genes on chromosome 16 that are orthologous to human chromosome 21 with none of these other things. So everybody said, why aren't you using duplicated 16? So we did. And we did probably what's the longest paper we've ever published. It's 12,000 words, but everything you ever wanted to know about these 3 mouse models, in which we compared them at 3 stages of life: in embryonic life, in neonatal life, and in adult life. And we looked at anatomy, gross histology, microscopic histology, stereology. We did transcriptome analyses of the brains, and we did behavioral experiments. And at the end of the day, what we showed was they were all different. They were not interchangeable at all. The so-called perfect model, duplicated 16, had no prenatal pathology. You couldn't tell the difference between them and their wild-type siblings. So not a great model if we're trying to evaluate the effects of prenatal therapy. TS65 was the most severely affected, and yet, again, the concerns that we had to pass it through the mother. And So we ended up with TS1-CJE knowing that it was milder in its effects, but at least it did have prenatal pathology. We then used our various transcriptomic analyses to identify candidate therapies. And so we took RNA from 9 tissue sources, mainly from our own work, but also from the literature. So back in 2009, we did an analysis of cell-free RNA in the amniotic fluid of fetuses with trisomy 21. We got information from the literature, from iPSCs and neurons, from fetuses with trisomy 21 compared to euploid fetuses, and then whole brain autopsy material from fetal cerebrum and cerebellum, those all the human side, and then we had all of our brain data from the various mouse models. We examined differentially expressed genes. You might say, well, why aren't you using sequencing here? And I'll explain why, because we were using the connectivity map data, which actually we needed to use microarrays, and we were just at that point in time evolving from microarrays to sequencing data. But we were basically looking for consistently dysregulated genes and pathways that were in common between the mouse and the human. So the way the connectivity map, it's a freely available resource from the Broad Institute. The way it works is you plug in your transcriptome, which is your biologic state of interest. In our case, let's say it was brain from mice with Down syndrome, and you're looking for how that transcriptome is affected by FDA-approved molecules. And they're using reference cell lines. And because RNA is a common language, you're using this either upregulation or downregulation of various genes in your data set or your tissue as it compares to the reference database. So you use that information to identify connections that tell you that certain drugs, FDA-approved drugs, are either going to improve the phenotype in theory or mimic the phenotype in theory. So we're looking for things actually that have a very negative signature, meaning they are negating the phenotype. And in a paper from Scientific Reports from our laboratory, we took these 9 tissues, again, in humans with Down syndrome and the mouse models, the embryonic brain from the mouse models, and came up with a list of drugs that looked promising. And why does it look promising? It's because the signatures are very negative, meaning it will, in theory, treat the dysregulated pathways. And the top candidate was apigenin, which is a natural compound that I'll tell you about in a minute. And if you look across, the majority of the signatures are negative. It's not 100% consistent. We were also looking for drugs that were going to be, in theory, safe to give to a pregnant woman. You can see that there are a lot of vitamins here. There have been some disappointing trials of using vitamins in Down syndrome. There have been some studies using metformin, but according to our data, metformin would not be, if you're going to use the connectivity map as your filter, that would not be our first choice. And we put fluoxetine on there as well because there are some trials going on with fluoxetine. So we again took our human data, we took our mouse data, we did separate connectivity analyses. The connectivity map is made for humans, but we did a variation of it for mice and came up with this list of 17 high priority compounds. And as I've already told you, apigenin rose to the top. Well, what is apigenin? Apigenin is 4'5'7" It's naturally occurring. It's in leafy green vegetables and citrus fruits, and it is known to have potent antioxidant and anti-inflammatory effects. In animals, it does cross the blood-brain barrier, and it has been studied in other situations where it has been shown to promote neurogenesis. So we're trying to improve learning and memory for children who have Down syndrome. That is part of their overall difficulty with development. And it has been used in humans. So there are 5 studies on clinicaltrials.gov. So having done this preliminary preclinical work, the hypotheses for the proof of principle study were that apigenin will rescue the clinical and molecular deficits in trisomy 21 and in our chosen model, which was TS1-CJE, and that the treatment would improve postnatal cognitive outcomes as measured by behavior, and that the treatment would lead to changes in gene expression that would essentially normalize the dysregulated pathways. So again, a very ambitious diagram, but we started with populations of trisomy 21 and gestationally age-matched and sex-matched amniocytes, and incubated them with apigenin at different micromolar concentrations to define the optimal dose, and came up with 2 and 4 micromolar concentrations were doses where we would see effects but not kill the cells, and then evaluated outcome using the COMET assay, which is a standard measure of oxidative stress, also measuring antioxidant capacity, and then looking again at the post-treatment transcriptomes. And then moving over to the TS1-CJE mouse model with their wild-type littermate controls, we mated C57 black 6 wild-type females with the TS1-CJE males, and they received a control diet, which was a powdered diet, and then the treated mice got the powdered diet with apigenin at a fairly high concentration derived from the literature, and again, evaluating with gene expression, with neonatal behavior using the homing test, where we put various odorants far away from the neonatal mouse and measure the amount of time that it takes for the mouse to smell the scent of their mother and rush towards their mother. And then we also did some adult behavioral tests, including the open field test, measuring anxiety, and the fear conditioning test, which measures memory, whether these mice remember that they've gotten an unpleasant foot shock. So here's some of the results. And they're on bioRxiv right now. They're under peer review. So they're at bioRxiv, so you can see it all there. But this is looking at the different doses of apigenin on trisomy 21 in blue and in the euploid cells in black, and showing that there's a very high amount of DNA in the tail, so it shows you visually here. The way that the COMET assay works is you've got cells that are stained with the fluorescein dye, and as they migrate across the gel, if there's oxidative stress, they progressively lose DNA as they migrate. So you see a tail that looks like a comet. So here are untreated trisomy 21 cells that you can see look very different from the untreated euploid. There are some comets there, but not to the extent that there are with trisomy 21. So you do see, even with 1 micromolar concentration, a reduction in the DNA in the tail. 2 micromolar was the sweet spot between reduction. You can see the difference between these two. You're losing that oxidative stress. You're also improving it in the wild type as well. So people often ask me, should all pregnant women take apigenin to reduce oxidative stress? I really don't know the answer to that. But in addition, fetuses with Down syndrome have very low antioxidant capacity. So it's a combination of oxidative stress and then not being able to deal with that. So the apigenin improved their antioxidant capacity. So at least the cellular level, it looks very good. We then ran through, and I'll show you the details of the experiments, but in mice with TS1-CJE, we first of all were very concerned about safety. There was no increase in perinatal loss or in seeing unusual birth defects in the dams that were exposed. And I should mention that the dams got their treatment. The minute the male was put into the cage with her, that's when the diet changed over. So they were on the diet from conception, since we couldn't really exactly pinpoint when conception occurred. Once they were put with the partner, that's when they got the diet. So no increase in birth defects. It definitely improved the performance of the homing test. In the adults, we saw some interesting findings that we didn't expect. We see a sex difference in males, not females. We only see it in the adults. We don't see it in the neonates. And we saw significant improvement in fear conditioning. As far as the gene expression experiments go, it's not a magic bullet, nor would you expect it to be. It does normalize gene expression in many, but not all of the genes. And how is it working? So I'm going to show you how we think it's working through a variety of upstream regulators. So just to orient you to the colors, wild type untreated is white, wild type treated with apigenin is gray, TS1-CJE untreated is orange, and TS1 treated with apigenin is red. Here are the neonates, and they have an abnormal hippocampal memory to start with. The apigenin significantly improved the latency to reach the goal. So what that means is it's the amount of time that they actually rush to remember where their mother's smell is. So there was a slight significant improvement in their olfactory memory. In the adults, it was really interesting to see the sex differences, and we are now seeing that consistently in the mice, and we think that something is happening probably post-puberty that's exaggerating the phenotype in males as opposed to females. And we don't know, it really hasn't been recognized in humans with trisomy 21. There's almost no literature on sex differences in humans with Down syndrome, so it's very interesting. And again, you'd only know this by studying males and female mice. Previously, like some of our collaborators that we've worked with have only worked with male mice, and we wanted to work with both male and female mice from the beginning. But again, if you look at the distance traveled, which is a measure of the anxiety and the hyperactivity that the TS1 mice have, the untreated mice are kind of running around their cage or in the box much more actively than the wild type. That is calmed by the treatment with apigenin. We don't see that in the females. In the females that are untreated, we don't see these differences. In fact, we see similar differences in the treated wild type and in the TS1-CJE mice. So apigenin is not necessarily therapeutic in the females. And importantly, with the fear conditioning test, looking at the hippocampal memory, here we're looking at whether a mouse remembers that it's been in a box previously and exposed to a shock. When you put the mouse back the next day, it should freeze because it should remember that something bad happened in this place. And you can see here that the untreated TS1-CJE mouse doesn't remember, it doesn't freeze. But when you treat it, if it's a male, it does freeze. So we are clearly improving the memory. I see we've lost some of the little asterisks look smaller, but it is statistically significant with the treatment. However, with the females, we just don't see it. So there is a sex-related difference. And so how is this all working? What is the underlying mechanism? And we think that it is working through upstream regulators. One of the challenges with Down syndrome is it's a very complex disorder. Multiple pathways are involved. It is not just the genes that are on chromosome 21. It's a global dysregulation. And so in looking at some of the, again, in the untreated human amniocytes, untreated mouse, we see that there are a variety of genes that are dysregulated, both upregulated, downregulated compared to controls that lead to pathways that are associated with increased inflammation. And that's certainly what we see in the histology in the brain of the mouse models. We also see that there is a dysregulation of cell cycle progression. And we're particularly interested in these because we see the same thing in the mouse model, which would allow us to see the effects of therapy. In the treated human cells, as well as the treated mice, you do see the reverse. So you do see consistent evidence mediated through the upstream regulators that there's decreased inflammation, as well as promotion of progression through the cell cycle. So we see that in the mice, we see that in the humans. We do see some additional findings in the mice that are associated with both neurogenesis as well as stem cell maintenance. So another aspect to explore in the mice. Now, it would be great if we could give apigenin to everybody, but again, as I've already said, there is a sex-based difference. So we think as a first cut, we would have to make sure that any therapies that we would propose to give in a human clinical trial work equally well in males or females, or identify treatments that work better in males versus working better in females. And this is just showing you 9 different cell lines of human trisomy 21 cells where, for example, in JL779, which is a trisomy 21 cell line, that particular cell line responds extremely well to apigenin, and I believe it's a male cell line. This line, however, which is also a trisomy 21, BB16-225, does not show very much proliferation. It actually responds much better to a different candidate therapy, which is DMOG, which is also on our list of candidate therapies to test. So I think this is why we need personalized medicine. And so in preparing for a human prenatal clinical trial, here's what the vision would be. I told you at the beginning, we now have outstanding screening in humans to identify fetuses that have trisomy 21. And this is what makes, if you're going to talk about treatment of children who have intellectual disabilities, what makes Down syndrome unique is we have a widely applied screening test and we have a gold standard diagnostic test. So if you were going to try to do this for autism, for example, you'd have the challenge that there are many, many different ways that a child can develop autism. You wouldn't have that one opportunity. So what makes this so exciting and so feasible is you can identify the fetuses that are potentially at risk for Down syndrome, and then we would recommend that an amniocentesis be performed to obtain cells from that particular fetus. Because remember, the amniocytes and the other cells that are floating around the amniotic fluid are fetal. They're 100% fetal in origin. So you can culture those cells, and that's your uniquely personal population of cells upon which to test your safe candidate therapies. And then you say, okay, well, for your pregnancy, because it's a boy, or you don't even have to tell her that it's a boy, but we're going to recommend that you take apigenin, so you offer that to the pregnant woman. And then we have a number of different early clinical endpoints. I'm just going to show you some work on fetal MRIs that we're doing. And then hopefully, and here's Michael again, hopefully after the treatment has been taken and we get through our clinical trials, that there is some improvement. Now, Tomo Terui, who's a former trainee of mine at Tufts, is funded by NICHD to study fetal brain development in fetuses with a number of different abnormalities, but in particular with Down syndrome, and he's just published recently on results from 10 living fetuses with Down syndrome and 13 controls. So this is very important for us because if you were going to do a clinical trial, you want to be able to measure what impact, if any, you're having on development of the brain. And most of the abnormalities are being detected in the third trimester, most notably the fact that the cerebellar growth is beginning to lag in the third trimester. But also, you can see even here, if you're not an expert in this area, that the shape of the brains are different and the way that the Sylvian fissure is oriented is different. So we are identifying landmarks in the MRIs of living fetuses that can help us establish whether or not there's an effect of therapy. Well, is therapy ethical? I think that's a really important question to ask, and if it is ethical, under what conditions? And I was very privileged to work with two quite prominent bioethicists in the Netherlands who were fascinated with this issue, and they did an analysis of women in the Netherlands, and it's very interesting. Women feel very differently about trying to optimize the health of a fetus who doesn't yet have a personality. They don't know the child yet. People feel very differently if you're talking about changing the personality of a child that they know. But they seem to be very positive about doing whatever they can to ensure that their child comes into the world with optimal health and development. And we've certainly seen the same thing in the fetal surgical world as well, that women, couples are very interested and highly motivated to do what they can for fetal therapy. And I would suspect for postnatal therapy as well, but here we're talking about actual potential brain changes. So I thought that was really interesting. The other thing is Dr. Google. I mean, for a long time now, people are not necessarily relying on what their doctor tells them. They're doing searches for themselves. And this happens to be from 2015, but even today, probably on a monthly basis, I get emails very similar to this one. This is a real anonymized email from a woman who was 19 weeks pregnant, and she just learned that her son has trisomy 21. She's a physician assistant. She's been scouring the internet. I want to take something while he's still in utero. I want to contact you. And people are asking, what can I do? They feel like, they know they're going to continue the pregnancy. They want to know this woman is on prenatal vitamins as well as prenatal DHA. I have no idea where she got that from, because that's not something that's certainly being discussed in trisomy 21 circles. So people are highly interested and motivated. So we're very excited. This is an incredible time to be able to be working in this area. And I'm on my summary slide. Thank you. Timing is looking good. So I hope I've been able to share with you today that gene expression studies using cell-free RNA in the amniotic fluid, I didn't really show you the reproducibility, but you can go to the literature to see that there are these characteristic patterns of dysregulation that are consistent with pathophysiology. We've used the connectivity map at least to identify FDA-approved drugs that can be repurposed to test these hypotheses regarding treatment. Our initial work with apigenin as a proof of principle has been, it's not perfect by any means, but it is certainly highly encouraging, and because it is safe and because it's a natural compound, it would lend itself to clinical trials. We think that it's achieving its therapeutic effects by reducing neuroinflammation and promoting the cell cycle, promoting neurogenesis. In vivo, it rescues the redox homeostasis and balance, and then in vivo, it improves behaviors, again, with this recognition of sex-specific effects in adult mice, particularly in males. So our future research is we are examining in more depth the pro-inflammatory cytokines before and after treatment. We're doing live cell imaging to look exactly how the cells are responding to the treatment and how it affects the progression of the cell cycle. And then we only gave the treatment in this study, we gave the treatment, from conception through whenever the experiment ended. We need to determine whether the in utero treatment is having more of the effect or whether it's the postnatal treatment. We also just picked one dose. We don't know if that's the right dose. And we are working now with a fourth mouse model, which looks promising to see, can we duplicate the effects in a second mouse model before going to a human clinical trial. Just want to mention, for those of you who are interested in Down syndrome, so it's kind of a happy circumstance that I arrived at NIH around the same time that NIH decided to significantly increase funding for research on Down syndrome. So I am co-chair of the executive committee of the Trans NIH Include project, which stands for investigating co-occurring conditions across the lifespan to understand Down syndrome. We have more than doubled funding available for Down syndrome. And the purpose is to investigate conditions that affect individuals with Down syndrome and the general population. So why do people with Down syndrome have a higher incidence of Alzheimer's disease? What can that teach us for everybody else who doesn't have Down syndrome, who is going to develop Alzheimer's disease? Or even at the placental level, the placentas of people who have trisomy 21 are senescent. They're prematurely aged, just like people with Down syndrome. So is there something that we can learn about the placentas that will teach us about placental abnormalities in fetuses with normal chromosomes? So there are three different components. One is these high risk, these transformative R01s that include basic science. We're assembling cohorts, large cohorts of people who have Down syndrome who have not been included in research. And then also these cohorts will be used to test new therapies. And so the benefit is improving quality of life for people with Down syndrome, but at the same time, teaching us things that relate to everybody else. So if you just Google NIH Include, you will find a lot more about it, but it is an area that is, we hope will be sustainable, at least for the foreseeable future. There's a tremendous amount of progress that's already been made, even in the first year. So I want to thank you for your attention. I also want to acknowledge the members of my new lab at MCHRI, as well as our bioinformatics colleague in the Netherlands, Jeroen, and Umadevi Tantravahi, who sends us a lot of our cells, as well as past and current funding. So thank you very much for your attention. Do we have time for questions? Yes?
Excellent presentation.
Thank you.
Could you say something about chimerism in Down syndrome and how is that influenced and have you studied that?
Yeah, so when you say chimerism, are you referring to chimerism in the mother or mosaicism?
In the fetus, not all cells will have trisomy.
Right.
Some will be normal.
Correct.
And at least for long-term infection, we looked at cancer, it made a difference.
Absolutely.
True trisomy.
Yeah, so that is a very, very important question, and especially when we're developing, so some of the work that we're doing now is developing iPSCs, and we are regularly checking the cells because even some of the ones that you get from Coriell are mosaics. So it's not just trisomy 21, there are euploid cells in there as well, and that's going to skew your effects. So you do have to consistently be aware of the fact that it's very easy to lose a chromosome. And the default is really to go back to the euploid state. So we are very aware of the issue and we regularly monitor because you wouldn't want to choose a therapy based on testing euploid cells. Yes, hi, Brian.
Great talk. Another example of an underdeveloped brain is extremely low birth weight preterm infant. And has epigenin been used in that domain? Because we know that all structures are underrepresented, particularly the hippocampus, the vermis of the cerebellum, and so forth. It would be exciting if this is a safe therapy that it could be used in postpartum treatment of babies who are actually fetal when they're born.
Yeah, so I hope everybody heard that question, which is whether apigenin has been used as a potential therapy in preterm infants with underdeveloped brains. And to my knowledge, it has not. It's actually never been used in a perinatal context. It's mainly been used in adults, either adult mice or adult humans. So I think that would be really fabulous if it also had applications elsewhere. You know, if it's safe, then potentially could be used for that. Yes, sorry.
Any strain difference between like the different mice? Have you tried like bulb C, B6?
Yeah, so the question is of the background strain, have we seen differences? And so we are mainly limiting ourselves to the Jackson Lab available strains, which most of them are on C57 Black 6, but there are limitations with that strain because they're very poor mothers. They cannibalize their young. So the TS65 is actually on a different background strain, and that's another confounder. But in general, we're staying with C57 Black 6 because we don't want to add in other confounders in terms of background strain. But it's a great question. It's just another place that you're going to get variability in the phenotype. Okay, thank you.